Usted me dice, doctor, para iniciar la grabación.
Sí, cuando quieran.
Ya empecé grabación, Jorge.
Ok.
Bueno, ya tenemos gente. Okay. So we will start. Well, good afternoon, everyone. We will continue with the second plenary lecture of the 17th International Conference on Electrical Engineering, Computer Science, and Automatic Control. This time as a virtual conference. And we have a great honor to present to our next keynote speaker, Professor Emilio Sacristan Rock, who is professor and director of the National Center for Medical Instrumentation and Imaging Research, CI3M, at the UAM-Iztapalapa in Mexico City, Mexico. He will present the next talk, Magnetic Stimulation of the Facial Nerve for the Emergency Treatment of Stroke. So you can share your professor, you can share your presentation. So I will give you a short introduction about Professor Emilio Sacristan. Professor Emilio Sacristan Rock is a well-respected Mexican researcher, inventor, and entrepreneur in the field of medical technology. He holds a Ph.D. in biomedical engineering and is professor and director of the National Center for Medical Instrumentation and Imaging Research, CI3M, at the UAM-Iztapalapa in Mexico City, and also founder of 7 medical technology startups. In 2017, he received the Mexican National Prize for Science and Technology in the area of innovation and design from the Mexico's president. He also is a member of the National Researcher System Level 3, and is a well-recognized researcher and engineer in the area of biomedical engineering. So thank you very much, Professor Emilio Sacristan, to accept to give us a lecture and to share your experiences, your technology, your inventions. Thank you very much. And you can share your presentation.
Thank you. Can you see it? I think I'm sharing it. Can you see
Yes.
the screen? You can see my presentation?
Yes, sure.
And can everyone hear me okay?
Okay.
Yes, we can hear you.
Okay, so I will begin. So, well, first let me thank the organizing committee, and thank all the attendees to this conference. I hope you'll find it interesting. It's a pleasure to be here. Let me start with explaining that this project, magnetic stimulation of the facial nerve as an emergency treatment for stroke, is a project that we originally started at the Mexican Center for National Center for Imaging and Instrumentation Research at the UAM-Iztapalapa. But eventually, after we got to a certain point and had the proof of concept, we created a startup as a spin-off called Nervive. And Nervive is a US-based company based in Ohio that is continuing the work in collaboration with CI3M to continue this work. And I'm here representing both. So I've been involved with this project from the beginning, and I will tell you the story of this project. So let me begin with discussing the unmet clinical need and the product concept that we're trying to develop. So the problem is stroke, and we're talking about brain stroke. Stroke is very common. There's over 16 million cases a year worldwide, 5.7 million
Professor,
fatalities.
Could you show your page in full size, please?
I'm sorry?
Could you show your presentation in full size?
It's not full size?
Is it in PowerPoint? The slides in full size.
They're full size on my screen.
Yes.
I don't know what you mean. This is as full size as it gets.
Okay, it's okay. Yeah.
Okay. And stroke is also the second cause of morbid mortality in the world, and one of the leading causes of healthcare costs, because patients that don't die from stroke will face long, lengthy recovery and rehabilitation. And treatment, there's basically two kinds of treatments for stroke. One is to try in the first few hours to restore the blood flow to the brain, but this is only effective if it's applied within the first few hours, 6 to 8 hours from the onset of stroke. If you can restore blood flow, you can save that part of the brain. The only other therapies that are available for a stroke after those first few hours is basically rehabilitation. The damage to the brain is irreversible. And then the only treatments available are to help in the rehabilitation, but that part of the brain will be lost. And there will only be treatment if the patient survives. The big problem is that less than 5% of patients can get to a hospital that can treat stroke, get brain images, get properly diagnosed and treated within that time window. We have to make sure that we can provide treatments that will restore blood flow fast in the first few hours, so that we can implement fast treatment protocols. Patients that do not receive timely treatment will face possible death or serious disability. So this is the problem with stroke. And I should point out that there's two kinds of stroke. There's a ischemic stroke, which is caused by the blockage of one of the cerebral arteries, which will affect the blood flow to a part of the brain that will eventually die. And the other kind of stroke is hemorrhagic stroke, which is where our vessel ruptures and causes a bleed within the skull. And this in turn causes the intracranial pressure to go up to the point where inside the skull, the outside of the brain, the pressure is equal to the arterial pressure, which means that then the blood can't flow into the brain, and then all of the brain is affected by low blood flow. So what is our proposed solution? Our proposed solution is to develop a medical device which is based on magnetic stimulation of the facial nerve. The facial nerve has been shown to have a vasomotor component to the nerve. The nerve has many functions. It has a motor function that allows us to smile. It has a sensory function that allows us to feel a slap. Also to feel our palate. So it also deals with taste, but it also has a component that regulates blood flow to the brain. And this is the part of the facial nerve that we're interested in stimulating, which has been shown that if you stimulate it, it causes vasodilation of the cerebral arteries, and it restores blood flow to the brain. And we have shown that we can, after a single 2-minute stimulation, we can significantly increase blood flow. We can up to double the blood flow to the brain after a 2-minute stimulation, and that this effect lasts hours, even possibly days, which will buy time and save brain, which would be of great benefit to these patients. One of the advantages is it does not require diagnostic imaging before treatment. It is relatively safe, so you could apply it to patients that are suspected of stroke, even if they have a hemorrhagic stroke. And it should be we should be able to apply it earlier in the emergency department or even in an ambulance before anything else is done. So as soon as a patient is suspected of having stroke, they could be stimulated for 2 minutes before anything else is done. And this might have a significant impact in the treatment of these patients. The idea is that the vital flow is an emergency treatment for stroke. An analogy would be that this would be like a cardiac defibrillator, but for the brain. And of course, it's not electrical, it's magnetic. But it's an emergency treatment that could be used at the earliest possible time to try and save brain tissue. And the way this works is that the facial nerve has many branches, but they all converge in something that's called the geniculate ganglion, which is just outside the cranium, right behind the inner ear. And from there, it goes into the brain, and it controls the arteries, the opening and closing of the arteries in the brain. So sorry, our strategy.
We can see your slides.
You can see the slide?
We can see.
It seems that we're having some internet problems. Maybe you can turn off your video so you can have a better connection, maybe.
Yes. Okay.
And also we lose the presentation. We're only watching your camera right now.
Okay, I turned off the camera.
Okay.
Can you see the presentation now?
Yeah, I think it's better.
Not yet.
Yeah, we can see it now, and it seems that everyone sees the presentation now. Okay.
And can you still hear me okay, or is my audio being cutting in and out?
No, I can. Yeah, we can hear you.
Okay.
Sorry for interrupting you.
So the idea is to find a way to stimulate the facial nerve at the geniculate ganglion just before the nerve enters the cranium, and this will cause a vasodilation. So one of the problems, of course, is that in a real patient that has not undergone diagnostic imaging, we do not know which side of the brain is having the stroke. And we have seen that stimulating on one side has a somewhat of a contralateral effect. So our strategy is to stimulate both sides of the head at the same time. So we will stimulate the geniculate ganglion on the right side and on the left side simultaneously to double the effect and to increase the blood flow in all of the brain. And for this, we've designed specific coils. I'll talk a little more about that, to specifically manage to stimulate the facial nerve at the geniculate ganglion. So this is how it works. So we place a coil, a magnetic coil, right on top of the ear and aligned with the ear canal. The ear canal helps us as a anatomical marker because the ear canal points straight towards the genicular ganglion. So we use the ear canal to orient the coil, so it's pointing straight at the genicular ganglion, and then we pulse the electromagnetic coil with short biphasic pulses.
Dr. Emilio, sorry to interrupt. We are having a little bit of problems with sharing your presentation. It could be possible that you stop your sharing and start again the sharing. And
okay.
Perhaps.
Okay, it says I'm sharing.
Yes. Yeah. Now I think that the problem is when you press on play and it becomes full screen, it is not refreshing to all. So perhaps it's better that you present in that mode.
Okay. Well, I'll make the screen as big as I can.
Thank you.
Okay, we can see you.
How do I hide this? Okay, does this work? Can everyone see this okay?
Yes, we can see it.
Yes.
Okay. Let me see if I can put this on slide only. Okay, maybe that's a little better. Okay, so this, can you see my cursor, my arrow?
Yes, we can see the arrow.
Okay, good. So this is the genicular ganglion, and we pulse the electromagnetic coil. We send these powerful pulses, biphasic pulses, that will go positive and negative, to try and stimulate the nerve right at the genicular ganglion. The fact that we have a 90-degree bend on the genicular ganglion helps, because what actually triggers the nerve is the electric field, the gradient in the electric field. And the fact that it has a change in direction allows it to be triggered a little more easier. Now, the genicular ganglion is about 4 centimeters deep from the coil, which means that we have to make these very powerful magnetic pulses. The magnetic pulses we're generating are 1.6 Tesla at the surface of the coil. And that's what we need to be able to achieve 100 volt per centimeter electric gradient at the genicular ganglion. So these are very powerful coils that we place along the ear canal. And we do repeated stimulation very fast, 10 times a second, to induce a current in the facial nerve and to get the physiological response. Now I can't change the slide. Okay, we'll go this way. And this idea of stimulating the facial nerve is not our own. It has been known for a long time that electrically stimulating the facial nerve causes vasodilation of the cerebral arteries. And in fact, there is a company called BrainScale in Israel that has already invested more than $350 million to develop a device for the treatment of stroke. But their device requires that the patient come into the hospital, is diagnosed with stroke. They have to go into the OR, where a hole has to be drilled in the palate, and a small implanted electrode is inserted to touch the nerve, so that it can be stimulated from a coil outside of the body. The problem with this device, of course, is that it requires assembling a complete surgical team, And usually this can't be done within the treatment window. BrainsGate wants to use a treatment window of up to 24 hours, which we believe is way too long. And they have just completed a large clinical trial worldwide with more than 350 patients. And they were just short of their efficacy goal, which was 95%. They were at like 94.8%. So they've been allowed by the FDA to continue the trial to see if they can reach that efficacy mark. And we expect that within probably very soon, we'll be hearing whether they get FDA approval. It's not really a true competitor because this is a very highly invasive device. But if they get approval, this would make things a lot easier for us because we will come up with a non-invasive way to do the same thing in a much more direct way and simple way that does not require surgery. So this is the basic idea of this device. Now let me tell you a little bit about the research we've done. Let me first start with our proof of concept in animal models. When we first had the idea that maybe we could stimulate the facial nerve magnetically, we used a technology that currently has been developed for stimulating the cortex of the brain for treatment of depression. And it's called transcranial magnetic stimulation. So we used this device that was designed for something else, but we tried to use it to stimulate the facial nerve. And our first trials, we tried it in sheep and we tried it in several different models. And eventually we developed a dog model in dogs with a clinically relevant model of ischemic stroke, where we inserted a catheter into the cerebral arteries, into the internal carotid artery, and injected blood clots so that they would plug up the cerebral arteries. And so these are brain scans that show this is the head of the dog, and you can see the brain here. This is a reference scan. And then we did a perfusion map where we can see where the blood is going. And this is the baseline before we did anything. We could see that this area, which is the brain, you can see that it's in bright orange. Bright orange means there's a lot of blood flow. Black means that there's no blood flow. And you can see that the brain is very nicely perfused under normal circumstances. Then we went ahead and injected the blood clots and caused a stroke. And then you can see in this area, we have a large black area in the brain, which is where the stroke has happened. And you can see a big infarct, big area of the brain that is no longer being perfused. Then we went ahead and stimulated, in this case, we were using transcranial magnetic stimulation devices, and we're only stimulating one side. We stimulated this side, and we immediately saw an increase in blood flow. But the interesting thing was that this actually increased over time, and even an hour after, we could see that the blood flow to the brain has not been restored entirely. There's still a large area of infarct here, but big areas of the brain have restored blood flow compared to right after the onset of stroke. And if we looked at the infarct size, we could see that the patients, the subjects stimulated had much reduced infarct size, in the order of 3 to 4% of the total brain volume was ended up dying. Whereas in the control group, which were the untreated, unstimulated dogs, the size of the infarct was around 22%. So that's a big difference between having an infarct that's 3% of the brain to 20% of the brain. I mean, that could mean the difference between being able to speak or being able to breathe or being able to walk. So that makes a big difference in the possible outcome of patients. So then we wanted to understand a little better how long this effect lasts. And we did several trials doing the stimulation and following it along over time to understand the effect of the stimulation. And here we have this was in pigs. Pigs in many ways is a better model for humans, except for the fact that there's no way for us to introduce a catheter into the cerebral arteries in pigs. Otherwise, we prefer pigs and dogs. In this case, these are normal pigs, and we took a baseline measurement of their blood flow or their perfusion index over the entire brain. And then we did a first stimulation, and then we measured the blood flow. We could see an increase of blood flow around 170 to 180% of the original blood flow. And then we did a second stimulation after 90 minutes. This is in red. And we could see that we could maintain that high level of blood flow if we re-stimulate it every 4 hours. We could also see that if we only stimulate once, the effect of the increased blood flow lasts up to 4 hours or more before it goes back down to, and this is in healthy subjects. In patients that actually have stroke, we have found that there's some intelligence in our own system that when we reopen the blood flow, restore the blood flow to the brain then maintains the arteries open. It recognizes that this is what it needs, and it seems to be that once we restore blood flow, the blood flow stays restored in patients that are actually having a stroke, although we need a little more research to understand this better. Now, one of the problems that we were also concerned about is what would happen if it's not an ischemic stroke, but it's a hemorrhagic stroke. The concern would be that what would happen if we stimulate a patient that has a hemorrhagic stroke? Would we cause increased bleeding? So we also had to do another set of tests this is again in pigs, where we would, using neuronavigation, use a needle to poke and rupture one of the cerebral arteries and cause a bleed in the brain. And then we could measure the intracranial pressure and follow the blood flow in the brain over time to see what would happen and what would happen if we stimulate. And what we saw is we did not see increased bleeding. The stimulation did not cause the blood vessel to rupture again. The hematoma stayed stable. The intracranial pressure did not go up. And we also saw that the brain perfusion index increased slightly, although it didn't last very long. And it may not be clinically significant, but at least we proved that using this technology on hemorrhagic stroke patients would be safe. It might actually provide some benefit. So then, once we were convinced that this technology would work, we started the development of a clinical system that could be used in humans. And we started this process in 2014. We started developing the human system for use in humans. And for this, we needed to develop a special coil that would effectively stimulate the genicular ganglion. But one of the big concerns in our conversations with the FDA was that if we used a standard figure-8 coil, which is what is normally used to focus the magnetic field for deep nerve stimulation, that we would also necessarily stimulate the temporal lobe of the brain cortex. And the temporal lobe of the brain cortex is where epileptic seizures are formed. So the big concern was that if we stimulated the cortex, that we may trigger epileptic seizures. So we spent a lot of time with simulations, the magnetic field, and developed a proprietary coil that uses 2 overlapping coils that produce a very asymmetric magnetic field that cuts off very sharply at the height of the genicular ganglion, but then is deep below it. So it's very deep in this area, but then it cuts off very sharply at this height so that it would not stimulate the brain, but would effectively stimulate the genicular ganglion. The problem is, of course, that the genicular ganglion is directly underneath the brain. So it would be very difficult to stimulate one without stimulating the other. But we came up with this design and we tested it. And then we came up with a stimulation sequence where we would alternate stimulating the left side and the right side by 50 milliseconds. The pulse is 280 microseconds, and it'll go up to 1.6 Tesla positive and then 1.6 Tesla negative in 280 microseconds. And then we repeat this pulse every 10 seconds, and we alternate from the left side to the right side. And the reason we're alternating from the left and to the right is we didn't want to induce currents in the opposite. Both coils will be facing each other, and we didn't want to induce currents in the from the stimulation on the left side onto the right coil, and from the left side to the right coil, and from the right side to the left coil. So we stagger them by 50 milliseconds, and then we repeat this over 2 minutes, and we ramp up from about 0.3 Tesla to about 1.6 Tesla, and then stimulate at 1.6 Tesla for 2 minutes, and then we ramp down again. And this is the stimulation sequence that we're using in humans. And this, of course, the power setting and the duration was done after we did a dosage response curves in humans, in healthy humans, at different powers and durations in a first trial. And this is what the actual device, our first prototype, looks like. We have 3 of these prototypes. We have 1 in Mexico and 2 in Cleveland, Ohio right now. And this is what the device looks like. And these are our proprietary coils that we developed and figure out how to build them and make them safe. They are cooled with silicone oil, actively cooled so that they won't overheat. Since we are using pulses in the order of 8,500 amps peak to peak while we're doing the stimulation. This is a very powerful magnets that we create to be able to go deep enough to stimulate the facial nerve. And when we tried this in humans, we tried it first in normal subjects. We tried it in 35 healthy subjects. And we did the 2 stimulation. And you can see here, this is the brain. This is a perfusion map. Again, the brighter the color, the stronger the blood flow. And you can see before stimulation what a normal brain looks like. And after stimulation, you can see a significant increase in blood flow throughout the brain. And we tried this in humans. And this is done with perfusion mapping in our MRI system. And we did this, and we also wanted to see if there were any adverse reactions to the stimulation for safety. So we wanted to make sure that we had the proper, and we ended up choosing 1.6 Tesla as our dosage for all patients. And this is the kind of adverse reactions that we saw. The most common one was a bit of jaw pain and soreness, because we can't separate the vasomotor component from the motor component of the facial nerve. So it makes the face twitch, and we make the facial muscles contract. And this caused some jaw pain and soreness during the stimulation, but that went away as soon as we stopped stimulation. The other thing that we did see was we're also stimulating necessarily the other nerves, and we're causing increased sweating in patients. And the other thing we saw was a metallic taste sensation. But there were no significant adverse events, no hearing loss, no damage to the vestibular apparatus. The patients recovered perfectly as soon as we stopped, and the stimulation was uncomfortable but tolerable. It's a little bit like being slapped continuously, but not too painful, and you can certainly tolerate it if you're suffering stroke. Now, you may not be able to see this, but here I have a little video, and I don't know if it'll work, but let me try it. This is this is an actual patient that has vasospasm. Vasospasm is a stroke-like condition that develops in patients that had a intracranial bleed and are recovering in the ICU. And this is a very deadly condition that has about a 75% mortality, and there's no treatment. So we decided that this was the best patient population to test this first, in patients that really have very few options and are in great mortal peril. And so this is a patient that had vasospasm. This is a patient at the National Institute of Neurology and Neurosurgery in Mexico. And we place the coils on either side of the face. And let me run let me see if I can run the video. Hopefully you can see it. And this is what the stimulation looks like. I wonder if, can someone tell me if you could see it?
Yes, we saw it.
Yes, we can see it.
Okay, so that's what the stimulation looks like. And this same patient's brain scans, and this is perfusion imaging using computed. This is using angiography in the angiography suite in the neuro ICU of the institute, and areas in black are areas that have proper blood flow, and you can see that there's large areas that I'm pointing with my arrow of the brain that have inadequate perfusion. They're white instead of black. Ideally, this should look all black. And here you can see there's a large area right in the middle of the brain that does not have, right here, that does not have adequate blood flow. And this is a patient who's suffering vasospasm. And this is 30 minutes after we stimulated, and you can see that a lot of the blood flow has been restored. We have much better, almost everything is black, and there's a much better perfusion of the brain. And this is what the actual blood vessel looks like. We have the vasospasm, we have a constriction of the muscle that gets irritated from blood products from the bleed, and going to spasm and close up, and you get this distinctive look of the artery where you have these occlusions. Looks like sausage links, and we have these occlusions of the cerebral artery. And after stimulation, the same patient, you can see the same, and you can see that the blood flow is restored. The arteries have opened up. And the results were very, very exciting. We had only 6 patients that we did at the National Institute of Neurology. We expected in these patients that have confirmed vasospasm, we expected a 75% mortality. All 6 patients that were treated recovered fully. We restored the blood flow. The vasospasm did not come back, and all these patients were released from the hospital within a few days with no significant cognitive deficit. When the FDA saw these results, they authorized us an emergency humanitarian use exemption, which means we are already authorized to use this device for the treatment of vasospasm in the US. And that would be our first application. Now, the problem with vasospasm is a very small market. It's relatively rare condition, but this could be a lifesaver for patients that develop vasospasm. So with these results, we start looking at our product development ideas and looking at commercial development of this technology. We're here. We have this first device in red. And this could be used for vasospasm, but we're also going to use this device for our first trial for ischemic stroke in the emergency department. So we are developing this first, and that maybe is 10,000 patients a year in the US and Europe. It's a small market, but it's a market that we already have humanitarian device exemption. And where there really are no options for these patients. But the big market where we really want to make our mark is for ischemic stroke in the emergency department. This would be patients that go into the hospital and are confirmed to have stroke that are treated immediately as soon as they come into the hospital to restore the blood flow. And this is a much bigger market. This is a $1 billion market right here. And eventually, once we develop this, we want to develop a more portable version of this device that could be placed in the ambulance so that the patients can be treated before they even get to the hospital. Because time is brain. The sooner you can treat and restore blood flow, the better the odds and the better the prognosis for the patient. So this is an overview of our entire R&D plan. We did our proof of concept in sheep and pigs. We did a ischemic stroke model in pigs and hemorrhagic model in pigs. I've already presented you some of these results. We also did a traumatic brain injury model in pigs. We had to do a study to see how this interfered with the standard of care. The standard of care is to use a thrombolytic called recombinant, it's called TPA, tissue plasminogen activator, which is a thrombolytic, a clot-busting drug that will dissolve the clots. That can also only be used if it's supplied within the first few hours. We wanted to make sure that our device could be used in conjunction with the standard of care, because we would have a problem, ethical problem, if our device required the physician to make a decision to use magnetic stimulation or to use the standard of care. We wanted to make sure that the doctor could use both. And right now we have completed these studies here, and we are, just yesterday, we got our reply from the FDA authorizing our first clinical trial in emergency treatment for stroke in the US in several hospitals in Ohio and Cleveland. So we're about to start this here. So where are we now? Our next steps, we plan to raise additional funding. We're going to raise about $20 million to complete the commercial product development, the actual device that will look like what I showed you in the first slide, which will be a lot smaller and specifically designed for this application, and to complete our pivotal trials for ischemic stroke and to launch the first product into market for the treatment of vasospasm. We hope to have this as a commercial device for vasospasm by 2022 and for ischemic stroke by 2024. This is what we're hoping for. And we're in the process of moving forward with clinical trials. Anyway, that is the end of my presentation. I hope you found this interesting. Let me take some questions.
Okay, thank you very much, Professor Emilio. So now it's time for questions for the audience. If you have any question, please go on.
All attendees can open their microphones now, so please go ahead with your questions. Or if you prefer, you can write down your questions in the chat.
Okay, I have one question. The research looks very interesting. Just at this time where we are in an emergency, in a global emergency pandemic by the COVID. But I was wondering about the maximum level of magnetic excitation for a human patient and the implications with respect to the blood or the nerves or the circulatory systems or muscles. But what really happens to the patient?
Well, can you hear me? Can you hear me?
Yes.
Yes. Okay. So one of the problems is the amount of power we need to do to stimulate the facial nerve will vary from patient to patient. Different patients have nerves that are more susceptible to being stimulated. Other patients have less of a threshold. Unfortunately, there's no easy way for us to tell whether we're adequately stimulating. I mean, we can see the facial muscles twitching, but that doesn't necessarily mean that we're effectively stimulating the genicular ganglion. And there's no easy way to determine what the threshold is from patient to patient. So we, based on our trials, we decided that 80%, more than 80% of patients would respond, and was the lowest we felt we could go that would still have the effect. Now, the effect is either you get the effect or you don't get the effect. There's no in between. But if you go higher than the minimum threshold you need to stimulate the nerve, then of course you will cause harder contractions, will cause more pain, it's less tolerable, and of course it uses much more power. So then we have more problems with cooling and keeping the coils working properly. So we wanted to go as low as possible while assuring that most patients would respond. And we figured that the actual threshold is probably around 75%, but we feel we would go to 80%, give us a little margin to make sure that most patients would actually benefit from this device. There is no other side effect. I mean, it's uncomfortable. You do get contractions. It can get painful. The coils are loud. They have a clack whenever they fire. So we actually have to put in ear protection to protect the eardrum for the patients. But there is no adverse event. We have tested this thoroughly, and we have found no adverse events beyond the stimulation. Once we stop the stimulation, nothing really happens, except we have an increased blood flow in the brain. Did that answer the question?
Yes, thank you very much. Is
Okay.
there another question, please?
We have a question in the chat from
Yes.
Omar Sandra.
Yes.
I'm gonna read it. Thank you for the presentation. It was pretty interesting. It seems that a full stimulation of the brain is carried out. Would it be better if somehow you can stimulate only the affected area?
Well, we're actually not stimulating the brain. We're actually stimulating the nerve that controls the blood vessels. And there's no way to make it specific. Basically, what happens is we have an active control of our brain arteries that open and very rapidly open and close our arteries. This is what keeps you from fainting if you stand on your head or do a flip. We are tapping into the system, and basically what we're doing is we're saturating it to the point where it just gives up and releases all the blood vessels, and the blood vessels just relax and open up, and we increase the blood flow. Now, the effect will vary from patients. Some patients, their natural condition, their arteries are very relaxed anyway, so there's not much that can be gained. But most patients, their arteries are halfway constricted under normal circumstances. So we release and relax the blood vessels. We increase the blood flow. Now, there's another reason why this is of benefit, because if the area that is affected has a blood clot, our device has no effect directly on the blood clot. We can't dissolve the blood clot. But in our brain, we have a lot of what is called collateral circulation, where arteries are interconnected together. So they branch out, but they rejoin in several places. So if you can't get the blood to that part of the brain directly because there's a blood clot, you may be able to get some of that brain and some of that oxygen the long way around, going through other arteries that come around and come back to that area. And that reduces the penumbra. So we want to basically stimulate all the brain so that we can increase the blood flow around the infarct area and not just into the infarct. Does that answer the question?
More questions, please.
We have another question from Cynthia Lourdes Toledo Pro in the chat. I'm going to read it. This is a very interesting development. how much more relevant is it now that stroke OPED is numbered due to COVID-19 infections? Do you think it's going to be more used in the immediate future? Thank you.
Well, I'm not sure that COVID-19 numbers have really increased. I mean, stroke cases have increased during the COVID-19 pandemic. We may be having some reporting issues there. Certainly, we would hope that this would become, we have the hope that this would become a standard of care, that every patient that has ischemic stroke that gets to a hospital, if the patient is still within the first few hours, they will get stimulated no matter what. And eventually that this will become a very fast emergency treatment. That's where we see it will have the most impact. Now, it may not help all patients, but as long as it helps most patients and it doesn't cause damage to anyone, it's relatively safe, this could be widely used. It's certainly a lot more less invasive and tolerable than a cardiac defibrillator. I would rather be stimulated with this device than across the chest with a cardiac defibrillator. And that's widely used everywhere. So that's what we're hoping for.
Okay. More questions, please? Okay.
We have another question in the chat from Omar Said Barcenas. I'm going to read it. Could this treatment have a preventive application on people?
Well, I don't know about stroke. It would be very hard to justify the stimulation for patients that are having stroke. It could possibly be used in patients that have aneurysm. By dilating the arteries, you reduce the tension on the wall, and it might actually reduce the possibility of the rupture. But I don't know if that's the case, and that would be a very tricky study to conduct, because the alternative is you actually cause the aneurysm to rupture, and that would be terrible for the patient. So I'm not sure. But for vasospasm, though, patients that might be suspected of getting vasospasm that are in the ICU, this may be used prophylactically. And in fact, the National Institute of Neurology and Neurosurgery are already suggesting that they want to use it in all patients, whether they have vasospasm or not, they might get vasospasm. They might want to stimulate just in case, because vasospasm is really deadly and there's very little you can do about it.
More questions, please? I have one more question, Professor
Yes.
Sacristan. And because you mentioned several times your collaboration with Ohio, Cleveland, and some team. And how do you do that collaboration? Or perhaps if how do you work with people from USA, and how do you think to apply it here for our medical national system? I mean, for the regulations and verifications of this kind of equipment.
Okay, well, there's several reasons why we went to Ohio. First of all is money. We could raise money to continue the development. To date, we've raised more than $10 million in the development. And the last clinical trials in the last stage of development, we got an NIH grant for $3.5 million. There are simply no funds like matching like that in Mexico. We just can't get that kind of money for this kind of development. Second reason is that the market really is in the US and Europe. There will be a market in Mexico, but patients that are actually treated for vasospasm, there's only a few hospitals in Mexico that actually do it. And for treatment for stroke, we're very behind the rest of the world. Patients in Mexico that are suffering stroke, there's very few. And of course, we have a system that does not spend on some of the really basic stuff. So spending on expensive equipment like this on an experimental device is difficult. Now, we know that the National Institute of Neurology and Neurosurgery, they want to buy 3 as soon as we can provide them with the units. They're very interested, but there's only a handful of hospitals like that in Mexico. So the market really is elsewhere. There will be, of course, since this is a Mexican project, there will be development in Mexico, but really what will make this commercially viable is to be able to sell in the US and Europe. And we're hoping that by within 16 months, we should be in multi-center trials in both in Europe and in the US for ischemic stroke. And that's the way we went. And now how I work with this, I actually took a leave of absence for the last 3 years and actually moved to Ohio to live in Ohio. I've just returned to Mexico now, but I'm still managing the team in Ohio remotely. But yeah, that's one of the sacrifices we have to make as academics. We have to sometimes have one foot in each side, no? One in the company, one in the university, and to keep things going and to further the project along. If I wasn't going to do it, nobody else would. So this is the only way to make sure that this happens.
Thank you.
Okay.
We have another question from Gabriel Vega Martinez.
Yes.
Gabriel says, the subject is more than interesting. Doctor, I suppose this intervention generates heat. How it dissipates the generated energy?
Yes, the cooling is a big problem for us. Our first prototype that we developed it so that we could go up to 2 Tesla for 5 minutes. And our coils overheated significantly. We have a very deep cooling system with cooling unit that cools silicone oil that is non-conductive, that flows and irrigates the coil and keeps it cold. Fortunately, we've now realized that 1.6 Tesla for 2 minutes is just as effective as 2 Tesla at 5 minutes. And the amount of heat generated is a lot less. So now we're looking into the possibility that we'll be able to use another more simple method of cooling the coils, but we're going to have to actually actively cool the coil. Right now, we're thinking of using a device where we'll inject a coolant prior and really cool down the coil just prior to and keep it cold for the 2 minutes of the stimulation. But it'll be a one-shot deal, and it'll significantly reduce the requirements of the device. It'll also make it smaller, but yes, it's a big concern. But right now, it doesn't overheat. You can do the 2 minutes, and we've tested this by independent UL and Intertech have tested this. We do not exceed 35 degrees Celsius on the surface of the coil during the 2 minute stimulation. And patient contacting surfaces are even less than, don't even go up to 25 degrees. So it doesn't overheat. But it was a major concern for us, yes. Thank you.
Mm-hmm. Is there another question? No? Then we would like to thank to Professor Emilio Sacristan Roch for this interesting and excellent talk about some development and on biomedical equipment, very promising equipment, and your research. Thank you very much, Professor Sacristan, for your nice talk. And okay, this time is a virtual conference, and we would like to.
Thank you. I will include in the chat my email.
Okay.
If anyone has more questions or would like to contact me for any reason, I think I just posted it. You can always send me an email.
Yeah.
Okay.
All right. Thank you
you
very much, Professor Sacristan. Thank you very much for the audience. And so, as part of the conference of the CCI 2020 conference We want to thank you for your presentation, your collaboration. Okay, so your mail is already in the chat. So thank you as part of the audience. We would like to thank you, and thank you very much for your attention.
Thank you, everyone, and thank you for your patience and your understanding. Thank you.
Okay, thank you very much. Thank you very much for the audience. So we will stop here, and we will continue with the technical sessions at 3 o'clock. So please take your lunch in your own places. And okay, so now we can close this plenary lecture. Thank you, Professor Emilio Sacristan, and greetings from Professor Ernesto Suaste, who is there in the audience, connected.
Thank you.
Sí, bueno.